Okay, now we're going to look at a chapter on fungi. Okay, this is syllabus details. We'll put this up at the end to check back that we've covered everything. Now we begin with the word eukaryotic. Eukaryotic means the fungus cell has a nucleus. So human cells, all animal cells, plant cells, we all have a nucleus. So uh, fungi is the same. It has a nucleus. Contrast this with the bacteria in the Monera kingdom. They didn't have a nucleus. They were known as prokaryotic. So fungi are eukaryotic. We can divide fungi into two types, into saprophytes and parasites. Saprophytes feed on dead organic material. Parasites feed on a living host. An example here, there's a parasitic fungus living and growing on a leaf. Another example of that might be potato blight. And here's a saprophytic fungus feeding on the remains of an old log, an old tree. Looking at a big way that fungus cells are different from animal or plant cells, they have a cell wall, but the cell wall is made up of chitin. So this outer cell wall of a fungus cell, chitin, that's very different to the cell wall of a plant cell, which is cellulose. The cell wall here is a type of polysaccharide called chitin. Chitin is normally found in the exoskeleton of insects. Okay, just to recap, saprophytes feed on dead organic matter. They're important as decomposers. So mushrooms will be a fungus that's a saprophyte. And parasites get food from a living host, often causing harm. Potato blight would be a plant parasite. Athlete's foot here would be a human parasite. So in all of these slides, I'm going to ask you to pause and to take down the notes that are there. So this is a slimmed down version of what's important in the chapter. Now we have to look at two examples of a fungus, two members of the fungi kingdom. So the first one is Rhizopus. So we have to study Rhizopus in a little bit of detail. Rhizopus is common bread mold. So there's a piece of bread and a mold growing on it is called Rhizopus. So if we zoom up on that mold, we'd see it's actually made up of threads called filaments. Some of these threads go along the surface of the bread. Some of them grow down into the bread. So they're for anchorage and for absorbing food. And some of the filaments grow up into the air. And the top, there's a football-shaped structure called a sporangiophore. Called a sporangia. So the upright stems are called sporangiophores, and the structure at the top is a sporangia. The sporangia can produce lots and lots, thousands of tiny spores for reproduction. So back over here. Okay, Rhizopus is a saprophyte. It feeds on dead organic material. So in this case, you have the organic material wheat that's in the bread. It has filaments called hypha. These are long continuous tube containing many nuclei. The individual separations between cells are broken down, ending up with a long continuous tube. The nuclei are haploid. That means they have a single set of chromosomes. Now the structure of Rhizopus. So you specifically have to know all of these words. Rhizoids are for anchorage. They grow down into the bread. Stolons for horizontal growth. They grow along the surface of the bread. Sporangiophores for upright growth, and they'll support the sporangia. The sporangia is the football-shaped structure at the top. The sporangia produce spores. Spores are single cells capable of dividing by mitosis, which is a type of cell division, to form a new fungus. So an individual cell that's capable of dividing to form an entire new organism. Okay, here's an example of a diagram from a Leaving Cert exam. So you have the horizontal stolons, the rhizoids that grow down into the bread, the sporangiophores that move upwards, they grow upwards, and the sporangia at the top. So the sporangia will contain the spores for reproduction. You need to maybe take down a diagram like this as well, please. Now we're going to look at the life cycle, how it reproduces. The simplest form of reproduction here is asexual reproduction, involves a single parent. That's quite simple. When the sporangia here will dry out, it withers, and when it does, it bursts open, and all these tiny spores are dispersed or blown away. Each one of those is capable of landing on a piece of bread or a piece of food and will divide by mitosis and start to grow. So that's asexual reproduction, a single parent. Another type of reproduction that happens in Rhizopus is sexual reproduction. This happens less often and is more of a response to danger for the Rhizopus, that the food is running out or it's too cold or there's some harmful condition. So it's a way that Rhizopus enters into survival mode. Now, there isn't a male and female Rhizopus, but they are chemically different, and so they're referred to in plus and minus strain. So we have the plus strain of Rhizopus growing side by side with the minus strain. When that happens, a bulge forms opposite from each strain. And remember, there was lots of nuclei inside the hyphae. Many of these nuclei start to go into the bulge. So the bulge becomes a cluster of nuclei, and that's called a progametangia. When the wall forms behind the cluster, it encloses all the nuclei in a little structure, and that's called a gametangia. So a gametangia forms when the wall forms and encloses the nuclei inside the bulge. So at this stage, we have a little cluster of nuclei enclosed in a gametangia. The next stage is fertilization. The gametangia fuse, so the gametangia from both strains fuse together. The nuclei fuse each other to form diploid zygotes. Okay, so you could have many fertilizations going on here, but in general, only one of them will tend to germinate to form a new hypha. A dormant zygospore is formed, which can survive poor growth conditions. So we have the zygospore forms over here, which would remind you of the endospore that bacteria produce for survival. Here we have a zygospore that fungi produce for survival. So the zygospore can remain dormant for a number of years until conditions are suitable again. Remember, this is a microscopic structure. In suitable conditions, the zygospore divides by meiosis. That means it goes from 2 sets of chromosomes back to 1 set of chromosomes again. So back to haploid again, and germinates to form a haploid hyphae to produce sporangia and spores. So growing from this, a new hypha with a new sporangia capable of producing new spores again. Just to remind you again, just to note again, hypha and Rhizopus is normally haploid. It normally has just a single set of chromosomes. The zygote and the zygospore stage are the only stage that are diploid. They have 2 sets of chromosomes. So to go from diploid back to haploid, you need to have meiosis. Meiosis will happen here just before the new hypha grows. A quick look at nutrition. Rhizopus is a heterotroph. All fungi are heterotroph. No fungus can make its own food. They're all heterotrophs. It must obtain its food from other organisms. Okay, now Rhizopus is a saprophyte. It feeds on dead organic material. It's not going to grow on your skin. It must grow on something dead. In this case, it's growing on soft organic material. It grows embedded in its food, so the rhizides grow right down into the food. They form root-like structures for anchorage and absorption. Digestion is external, so enzymes are released onto the food. Digestion happens outside of the fungus. So the starch is broken down into simple sugars, and those simple sugars are absorbed into the rhizides. So digestion is external and the nutrients are absorbed. So this diagram here, you can see the food absorption going on down here, and you can see asexual reproduction happening up here with all the spores being released, each one capable of growing into a new fungus. So this can happen in a matter of days. So the fungus, once conditions are right, can grow very rapidly. Now we were looking at another type of fungus called yeast. Yeast is a single cell fungus. Okay, you've heard of baker's yeast and brewer's yeast. Okay, some yeast species are parasites; they feed on a living host, and others are saprophytic. They'll quite happily grow on organic material, dead organic material. So yeast is a single-cell fungus, like before the thin wall of chitin. The cytoplasm is quite dense. That means under a microscope it looks quite speckly because of the small uh, particles of glycogen are there for storing food. So there's a lot of little particles of food being stored here in the cytoplasm. Sometimes they'll have a small nucleus or a small vacuole. Okay, yeast get energy from food by anaerobic respiration. Okay, many yeast can respire anaerobically without oxygen, and if they do so, they'll produce waste gas of ethanol and carbon dioxide. So baker's yeast produces ethanol and carbon dioxide, and the carbon dioxide is what makes the bread rise. Brewer's yeast, again, produces ethanol and carbon dioxide, and it's the ethanol we need when we're making alcoholic drinks. Now, looking at asexual reproduction, a single parent, is by a method called budding. So we can see in this diagram we have a daughter cell growing out of the parent cell. And down here, the daughter cell is forming, it's developing even further, and there is an entire daughter cell beside it, and another daughter cell that maybe has developed from that, and another one that's developed from that. So you end up with a chain of daughter cells. Sometimes these daughter cells separate to form new yeast cells, but as long as they're still attached, you'd call them a daughter cell. Okay, so this is a characteristic feature of yeast cells, that they'll have a daughter cell attached, and this is why I've drawn the bud or the daughter cell attached up here. So asexual reproduction is by budding. Okay, quick description of that. The parent cell divides by mitosis. Mitosis is a type of cell division that produces identical cells. Okay, one nucleus enters a smaller daughter cell or bud. Now, we would have already mentioned this going through, but just to recap, in what way are fungi beneficial and what way are they harmful? Beneficial fungi are useful because they can produce antibiotics. The first antibiotic, penicillin, was extracted or discovered accidentally from a fungus similar to the fungus that grows on oranges. And we have edible fungi, they're used as food. Okay, so two good examples there. Fungi can produce antibiotics, and fungi are a source of food. And just to mention over here, we also have fungi are important in decomposition and the recycling of materials. So they break down the dead leaves and the dead organisms, so that the elements can be reused in nature. So they're important decomposers. Harmful fungi. So some mushrooms are poisonous, we've mentioned that. Fungi can cause food spoilage, so bread mold is causing food spoilage. And diseases like athlete's foot um, for humans is uh, obviously harmful. Plant diseases, just mentioned on there, potato blight is a plant disease. And many fungi can damage buildings. If the timber in buildings get wet, it can lead to fungal growth, which can cause wet rot, which is a fungal damage of um, timber in houses. Now, to move on, we need to do an experiment at the end of this chapter in this topic. It's a simple experiment, and this is a very simplified description of it. So we're looking at the growth of leaf yeast, which is a type of fungus that grows on leaves. Okay. Um, okay, so we want to grow leaf yeast. We don't want to grow anything else. We don't want to grow any fungus that might be on your hand or on the bench in the lab. So we have to take steps to avoid contamination. Okay, so collect the leaves from an ash in a bag. That avoids contamination. We need to know what the name of the leaf that we're using, so we're going to use ash leaves. Disinfect the bench with some disinfectant like Milton, and the scissors and tweezers can be sterilized in alcohol. So we're doing our best to keep everything free from contamination. So basically what we're doing is we're using sterile scissors to cut the leaf into small sections. You can see them here. we're attaching them to the lid of the Petri dish. Okay, use a little bit of Vaseline to stick them to the lid of the Petri dish. The lower surface should be facing away from the lid. Place the lid over a dish or Petri dish of sterile nutrient medium such as agar. Malt agar is specifically has all the food that the fungus needs to grow. So agar is a type of jelly. Nutrient agar and malt agar are examples. They contain nutrients that microorganisms need to grow. Okay, place the dish in an incubator at 25 degrees Celsius for 3 to 4 days. This is the time period needed for the yeast to grow. And if you're successful, you'll see pink colonies, okay, a definite pink growth, and you can just see them in the picture here. Now we're looking at the Petri dish from the bottom in this, so we're looking through the malt agar, it's transparent, and we can see the pink spots, and we can see they're lined up to where the leaves are. So these cells would have fallen from the leaves onto the agar and started to grow. As a control, place a Petri dish of malt agar in an incubator without leaves, and that will test how sterile your bench is and how successful your steps to avoid contamination have been. So the control should show no pink colonies. So the number of colonies you get will be an indication of the number of leaf yeasts that were on your leaf, and it might give you an indication of how clean the air was. Leaf yeast doesn't grow that well in towns because of pollution from cars. So plenty of leaf yeast would indicate that the air is pollution free. So what do we do with the dish at the end? We can't just throw it in the bin because it's a potential source of harm, of contamination for somebody else. So dispose the dish by burning them is one option, or soaking them in disinfectant. So a basin of a disinfectant like Milton, put all the dishes in to disinfect them, to sterilize them. Okay, finally, we want to look at what are the general safety precautions you should take when you're working with microorganisms. So this is the hazard symbol for microorganisms. It's kind of a warning sign, contamination. And two words that are important we should know. Sterile means free from all microorganisms. That would be important in a hospital operating room, an operating theatre environment. And asepsis free from pathogens. There may still be microorganisms present, but there's no pathogens present. That would be important maybe in a kitchen food preparation area or a doctor's surgery. Okay, so aseptic means free from pathogens that cause disease, and sterile means free from all microorganisms. It's more difficult to be sterile all of the time. You wouldn't necessarily boil your kitchen knife every time you use it. That would be trying to be sterile. But putting it into a dishwasher using detergent to clean it is relatively aseptic. So aseptic techniques: wiping the bench with a disinfectant like Milton, okay, to remove any harmful bacteria. Sterile techniques: okay, you might boil scissors or tweezers in boiling water, or use alcohol to kill all microorganisms. Pressure cookers can be used to sterilize glassware, to sterilize agar before it's poured onto the Petri dish. Containment. Okay, how are you going to prevent the microorganisms from spreading? So they're grown in suitable containers such as Petri dishes that can easily be sealed with parafilm. Parafilm is a special tape used to seal the Petri dishes. Obviously, we're not going to grow microorganisms in open Petri dishes. And disposal, what do we do at the end? We can't just throw them in the bin. They could contaminate somebody else who handles the bin. So we can soak the agar plates in disinfectant before disposal. It's generally a good idea when we're working with microorganisms to consider all microorganisms as if they are potentially dangerous. Now, finally, we're going back over our syllabus requirements and again to make sure we've covered everything. Eukaryotic means the cells have a nucleus. Saprophytic, okay, feeding on dead organic matter. Parasitic, feeding on a living host. Poisonous fungi can give you a stomachache. Sometimes it can be fatal. Rhizopus, the structure. Okay, you have the rhizoids for anchorage, the stolons on the surface of the bread, the sporangiophores to hold sporangia. The life cycle, we had asexual and sexual. There's a bit of detail in the sexual reproduction that's important to go back over that. Nutrition, we mentioned up here, saprophytic and parasitic. In specific, when it comes to yeast, is an example of a single-celled fungus. And it's important, you should know about budding. Budding is where the daughter cell is formed. The economic importance of fungi. So when we were talking about yeast, we would have mentioned baking and brewing. So they're economically important. Fungi are also important because they produce antibiotics. Harmful fungi, we would have mentioned poisonous mushrooms and the fact that fungi such as athlete's foot can cause human disease and potato blight can cause plant disease. This is our experiment description. Okay, all the steps, we use ash plants, we use malt agar, an incubator, we use sterile techniques, we identified pink colonies, and the control had no colonies. And then you sterilize the petri dish in a disinfectant at the end. We've more or less covered all the laboratory procedures in that. It's important that you know the difference between aseptic and sterile. Aseptic, free from harmful microorganisms, and sterile means free from all microorganisms. And again, we refer back to disposal is don't just throw them in the bin. We need to disinfect them with Milton or bleach before we get rid of them.